Hello, my name is Ashima Hassan. I'm a master's student at St. John University. Today I'm going to present my poster to I'm going to present my poster for the final assignment of communication skills in science. I'm going to talk about what we can say a trending genomic editing tool called CRISPR-Cas9 and how that CRISPR-Cas9 was able to reach to the Nobel Prize and how it could be used and applied for human being and how it was discovered or generated. Actually, if we would talk about Nobel Prize, it mostly occur at October of every year, where people elect people from different science and research fields or different life aspects were awarded the Nobel Prize. At 2020, the last year, the Nobel Prize at chemistry was awarded to both scientists Emanuela Charpentier and Jennifer Doudon. Emanuela Charpentier, she is a scientist at Max Planck Institute, and Jennifer, she is working at California University. They were awarded the Nobel Prize for generating and the usage of the CRISPR-Cas9 tools and how they will be able to use it in some human cell cultures to edit and alter some gene mutations or some gene defects. There were a big concern about CRISPR-Cas9 and if it's possible to use it in human being. And unfortunately, there was only one trial that was announced or was obvious to be known. That one was done by some scientist, Chinese scientist, he known as who HU, he was able to edit the gene responsible to produce some protein that would be recognized by HIV virus. Upon that, he claimed that he did his trials on human embryos for the sake of their survival, especially that the parents were suffering from HIV problems. But actually, according to the scientific committee ethics and rules, he couldn't perform such trials on human embryos. As on the long run, there would be big problems related to that. He claimed that his trial was successful from the first time, but the concern would be about if the genetic engineering trials will not be able to reach to a successful point. What he or what people or what scientists would do with the unsuccessful trials that were done on embryonic cells or human embryos. Actually, the Chinese scientist, his name is Hu, he got jailed for 3 years and his lab was closed and locked down, and also those who shared at the trials were put in jail, and they were they paid about, I guess, $250,000 something like that to the committee and to the society as a kind of penalty. If we talk about CRISPR-Cas9 application, it was a usage tool related to cancer, especially neuroblastoma, tumor genicity, functional and application of CRISPR-Cas9 will be mentioned by the design strategies and most significant results. We would talk also about the comparison between CRISPR-Cas9 and other editing tools. Actually, CRISPR-Cas9 is considered to be one of the most effective tools because what we can say, the Cas9 protein nucleases would be able to recognize lots of sequence on a human DNA. If we talk about the discovery of CRISPR-Cas9, actually it was CRISPR-Cas9 was discovered accidentally by the Japanese scientist Yoshimoto Ishino, and while he was trying to isolate some genes from E. coli in human microbial gut, the gene is ABI, which is responsible for the survival of E. coli in our gut, he found that there is a kind of unknown DNA around the ABI gene locus, and he compared that locus to other species from E. coli, and he found that this unknown DNA sequence is conservative, and it seemed to be to him that it does not coding protein at that time. That was in 1987. Forwardly, the Spanish microbiologist Francisco Moje coined the term CRISPR-Cas9, and he isolated the same sequence from different strains of bacteria, and he found that those sequences are conserved among different strains of bacteria. For instance, if it's E. coli, it would be conserved at that genome locus, if it's Salmonella or whatever bacteria. So that sequence are conservative, and they don't turn on unless the bacteria is exposed to attack by a virus or put under stress. So he put the term CRISPR-Cas9, which is cluster regulatory interspaced short palindromic repeats, because it's a short sequence and they are palindromic repeats and they are regularly or they found and conservative among bacteria. Then we talk about Wing. Is a Chinese scientist. He's very promising. He had a lab at Harvard University. He used successful CRISPR genome tool to edit human cells. He was able to edit some human cell cultures in 2013, and he was able to design and increase the efficiency and efficacy of CRISPR-Cas9 system. Then we talk about our Nobel Prize, who are Emanuela Charpentier and Jennifer Doudon. They were able to describe the process where the bacterial enzyme named Cas9 used to cleave invading pathogen and combined Cas9 with cystine single guide RNA and successfully cut the target bacterial sequence, meaning that they were able to isolate and distinguish the sequence on bacterial genome that could code for the Cas9 protein nucleases, and they were able to code that protein, and also they linked that protein to synthesized single guide RNA. So when that gene responsible for the coding of Cas9 nuclease was turned on, together with the synthesized guide RNA, they were able to cut the invading genome or a certain genome of the bacteria. Here they were able to cleave the invading pathogen, and they use such technology, which is the gene that turn on Cas9, together with synthesized guide RNA in order to be able to cut the pathogen genome. If we talk about the CRISPR-Cas9 as a bacteria vaccine, meaning that it's a kind of adaptive immune system for bacteria. So once here the bacteriophage attack the bacteria, it would insert its genome inside the bacteria. Then the bacteria would use the genome and insert it on the CRISPR sequence that would code for the Cas9 and also for the guide RNA. Here, the Cas9 is coded by the bacterial genome and also the guide RNA as well. Accordingly, they would have the tools that they would be used again in case if another attack would happen. So first of all, the attack of the bacteriophage virus would occur, and then the genome of the DNA of the genome of the attacking virus would be integrated into the CRISPR-Cas9 system that would encode forwardly for the Cas9 nucleases and guide RNA. So in case if another attack would happen, the bacteria would be ready. The point that we are using the same concept, but here the pathogen or the what we need to edit would be the foreign genome. If we talk about the components of CRISPR-Cas9, actually it composed of Cas9. This is a nuclease, and this is the guide RNA, the spacer. The guide RNA would guide the Cas9 to our target sequence, and the Cas9 would eat the preferred DNA sequence. Actually, there are lots of regulations for that. You need to be very specific about the guide RNA. You need to design guide RNA with higher efficiency and higher specificity as well. In case if it lacks specificity, the Cas9 could cut any place on the genome and then the forward or the consequence of your experiments would not be accurate. So if we need to knock out of the genome, we have to perform the Cas9 nuclease, would target our sequence. Then this is genomic DNA, then this is the guide RNA that would cut post strands, and accordingly for the DNA repair system would repair the muted or the cut DNA through either homologous DNA repair or non-homologous DNA repair. We can use that to correct mutation or to cause mutation to our genome or to complete removal of target genes. If we talk about CRISPR-Cas9, if we talk about the CRISPR-Cas system within COVID, actually there were lots of trials related to the usage of a CRISPR-Cas system to identify and cleavage COVID. The Cas nucleases here was Cas13 because actually Cas9 couldn't identify the RNA, so better to use other strains or other species of the Cas nuclease protein, which is Cas13. Actually, it was found that the CRISPR-Cas13 was efficient in identifying and cleavage of the COVID RNA in human lung cell cultures. It reduced the efficient, or what we can say, reduce the infection by about 90% in the human lung cell cultures. And some people were claiming that one day they would be able to use the CRISPR system as a kind of vaccine against lots of viruses and lots of foreign attack. But the point that there must be lots of trials in order to ensure that the CRISPR-Cas system would only target the pathogen and would not target any RNA or any DNA in the human being. The future of CRISPR-Cas9, it can be used in agricultural crops and animals to increase their productivity and to give the animals more strength against external pathogen, and what we can say for plants and crops like pests, and increase the productivity of agricultural crops and animal products. Also, it can be used in synthetic biology, like the usage of vaccine, or there could be also usage together with PCR to increase the efficacy and accuracy of the PCR product. It can use with ecological studies, like some trials were done to prevent mosquito from carrying the malaria protozoa and causing that virus. So there were some trials to knock out that portion of the mosquito genome that could be recognized by the virus. So if the mosquito would bite somebody, it wouldn't harm him or her. Also, screening of the drug and viral gene therapies and pathogen gene destruction. We can study how to destroy the viral genome, how to increase the immunity against such viral genome. The models for that would be either human being in case of cells, or also organ transplantations that could be done by find the similarity between human and other animals, and then we remove the different genes and we incorporate or we knock in the similar genes and then transplant that organ in other animal models like pig, chimpanzee, and then after the organ is completely mature, we can remove that organ and retransplant that, or put that to the human being. Also, to preserve some species of organisms who we could also use CRISPR-Cas9 to preserve some organisms and some animals who were extincted through either fortify the genome of their offsprings or try to get what we can say, the bad portions, or to remove the mutated portions that expose them to be extended through CRISPR-Cas9, and we can also lock in other favored genes that would give them more strength and make them more adaptive to the surrounding environment. At the end, there are still lots of experiments and trials that are going to be done in order to be able to use CRISPR-Cas9 as a safe genome editing tool, especially that some people consider genome editing tool as a whole as a kind of a bioterrorism tool. You can invade people now with diseases, you can invade them with microbes and viruses, like some people till now consider the COVID virus is a synthetic virus due to its high rate of evolution and its high rates of adaptation. Actually, we got more than 4 strains in less than 1 year. So some people think that it could be the bio weapon, and no one know where is the truth. But at the end, it's very obvious that CRISPR-Cas9 is going to be a very promising tool in genome editing, at least related to improving the environment, improving agricultural crops, improving animal health, and also what we can say, improve the human health, especially for such hopeless cases of cancer or genetic diseases related to thalassemia or Turner syndromes, or lots of what we can say, autoimmune diseases like Hashimoto. So CRISPR-Cas9 can be used for the sake of organizing and creating at the architecture more safe and more healthy individual. At the end, I would like to thank you for your kind listening. I hope I will be able to deliver the message very clearly, and I'm very happy to get questions from any one of you. Thank you.